Hi, my name is Marissa Williams, and I'll be talking about the sliding filament theory. So the first step in the sliding filament theory is when the myosin heads split and ATP becomes reorientated and energized. And then once the myosin heads bind to the actin, it forms a cross bridge. And then from that cross bridge, the ATP is used and the myosin heads rotate towards the center of the sarcomere, and that's called the power stroke because it's pulling them towards each other. So the myosin heads then bind to the ATP and the cross bridges detach from the actin, and then the cycle can start over again. So the contraction cycle continues if ATP is available and calcium levels in the sarcomere are high, Ca2 levels. So in my diagram, you can see the different stages of the cross bridges and the power stroke, and then the detachment, and then back again with the when the ATP splits, and then they lose ATP, and the cycle starts over again. So just to go more in depth on the process, the excitation of the muscle is the nerve impulse which is sent down to the motor neuron, and then it arrives at the synaptic knob, which causes the extracellular calcium ions to move by facilitated diffusion into the synaptic knob. And then the calcium causes the synaptic vessels in the synaptic knob to release ACH into the cleft. And then once the ACH molecules diffuse across the synaptic cleft and bind into the receptors on the sacromella, the binding generates the electrical signals, and then they speed into the sacromella, and that's how we know that the muscle's ready to contract. So after these signals travel to the cell surface down to the sarcoplasm through the transverse tubules, the impulses in the transverse tubules cause the terminal cisternae of the sarcoplasmic reticulum, and then it releases the stored calcium. So the stored calcium ions then diffuse, and then they bind to the troponin molecules on the thin filaments, and then the calcium and troponin bind to the tropomyosin, and then it pulls the myosin away from the binding sites on actin. So the myosin heads use ATPase to break down the ATP, and that's how it's able to release energy. So the myosin heads use ATP to extend the actin and attach to the binding sites on actin. So the myosin heads pull opposing thin filaments to the center of the sarcomere, and then the extra ATP binds to myosin. So after the ATP binds to myosin, they release the original cross-bridging, and then the ATPase breaks down the new ATP, and then the process repeats until the sarcomere shortens all the way, and that's the full contraction. So on this next image, it shows the relaxation of the muscle, where the actin is on the sides, not touching anything, and the myosin, the thick filaments, are in the middle. And so after the contraction starts, that's when the cross bridges are built by the myosin from the ATP, and then when the contraction finishes, it's shortened all the way where the thick filaments and thin filaments are all overlapping.